Okay, so I'd like to welcome everyone to this today's lecture on the Signaling Pathways Project, or SPP, component of dkNET. Hopefully, you've all had the opportunity to look at some of our recent articles. There were links in the handout that Kowai sent earlier, and what I'll be discussing this afternoon will make a lot more sense if you have. But anything that's not obvious, or that you're a bit hazy on, we can discuss that in more detail at the end of the lecture. So I want to walk you this afternoon through three real world stories that I feel best exemplify the way in which SPP analysis can be used. And the kind of overarching theme here will be SPP's investment of effort in making full use of existing data sets in a way that adds value and insight to investigator research. So I wanted to start by explaining what we at SPP understand as signaling pathway. This is a phrase or a concept that is unfortunately not very well defined. I've heard a lot of different kind of perspectives on what it means, and so I wanted to explain firstly how we here at SPP use this, or kind of view this concept. And as we see it, the term signaling pathway refers to a series of functional and physical interactions that basically actuate the cellular response to a signaling event. Now, this signaling event may be, a hormone or some other signaling molecule. It may be an environmental substance, or it may be some kind of interaction within the extracellular matrix. So it's really any event at the surface or inside a cell that initiates a response from the cell will activate a signaling pathway. And cells respond to signals in multiple ways, but arguably, at least from the kind of SPP point of view, are those that occur in the cell nucleus, and specifically those whose endpoint is the regulation of transcription. And depending upon how far along you are in your education, I know we have some people who are more senior students than others, but you may have come across this kind of a depiction of cell signaling, maybe as a figure in a review. And what these figures almost invariably focus on are what we refer to as the nodes in these pathways, which are the molecules whose physical and functional interactions basically transmit the signals from the cell membrane into the nucleus. And the three major sort of categories of signaling nodes are the receptors, which initiate the signal, and then a series of enzymes which amplify the signal, and those then sort of transmit the signal to the third major flavor, if you will, of signaling node, which are the transcription factors. But what's often marginalized in figures like this here, which you can see here in this particular example, is the impact of these signaling pathways on the regulation of expression in the nucleus. There may be a kind of a vague allusion to regulation of a specific cell function, but there's often very little in the way of the kind of a detail that the rest of the pathway is outlined in. So, as those of you who are active in research labs will be aware, most of our knowledge of cell signaling pathways is obtained from the research literature. It's often in the form of individual research articles or in reviews, which can aggregate facts from multiple research articles into a single article, and which often have a whole bunch of information, and then at the end they have a very kind of a complex sort of a schematic like this. However, there's another universe of information that informs upon cell signaling pathways, and that's in the form of archived omics data sets. And so again, for the purposes of SPP specifically, we're going to focus on two specific types of omics, and those are transcriptomics, represented by expression array and RNA-seq. There is more and more RNA-seq, coming out, but expression arrays are still used. And the other main sort of a category of archived omics data sets are what's referred to as cistromics, and the most common form of this is ChIP-seq. So what are these omics? Are these omics platforms, and what information do they give us? So looking at our kind of a basic representation of a cell here, for any given signal, your array or RNA-seq experiments will basically survey over thousands of mRNAs in a cell, and they will indicate which ones are increasing or decreasing in abundance in response to that signal. Whereas on the other hand, cistromic experiments will actually indicate which specific DNA binding factors are involved in response to a specific signal. And so, although these experiments, ChIP-seq experiments will not give information on which mRNAs are up and which are down, what they do indicate is which nodes might be involved, or that actually gives rise to that increase or a decrease in a specific mRNA. And so what's important to remember is that a specific signal can have multiple mechanisms of action. And so by combining array RNA-seq and ChIP-seq data, SPP enables researchers to identify the specific mechanism that might be mediating a specific signal in their particular system of interest. Now, even though omics platforms inform upon thousands of specific events in a cell as a consequence of the way in which research is done and is funded, what often happens is that in order to actually publish a research article, investigators will only focus on a small handful of those events that are of most interest for them, and they'll kind of look at those in more detail. And so what happens is that there's all this other information in their array experiment or their type-seq experiment that will not make its way into the research article. And even though that information wasn't of immediate interest or relevance to that specific lab, it might be of interest. Actually, might be of highly significant interest for other researchers. Now, although these data sets can be archived, that's still it doesn't happen all the time. And even when they are archived, the actual archived data sets can be very difficult for the average researcher to make use of. So the goal of SPP is to reuse existing archived omics data sets to basically make connections between signaling events and those downstream transcriptional targets that they regulate. And I want to introduce now the first of a couple of analytical platforms that we use. The first of which we refer to as consensomes. And as the name indicates, what consensome analysis does is to survey over multiple independent expression array or RNA-seq data sets, and it will then rank the transcriptional targets by the frequency with which they are differentially expressed over these independent data sets. Now, these aren't simply any old data sets. They are what they have in common is that they involve some kind of manipulation of a member of a specific node family. And I will that's the first kind of a consensome. The other type of consensome is the type-seq consensome. And here, what we're doing is we're looking over multiple independent experiments that all have a common IP antigen. For example, FOXA1 or estrogen receptor alpha, and we then rank the rank the targets by the mean peak height, which basically indicates the confidence with which a particular peak for an antigen can be identified. And so again, we are ranking those downstream targets by the strength of their relationship with a specific node. So I'll give you an example here. And I mentioned earlier that the consensomes for expression array and RNA-seq data sets involve manipulation of members of a specific node family. So I'll take the insulin receptor family. So this is the insulin receptor itself, but it's also IGF-1 receptor and a couple of others. But what we do here is we identify archived experiments that involve some kind of manipulation of a member of that family. So it can be, as simple as insulin receptor itself. It can be, loss of a function of the insulin receptor. It can be a ligand for another member of the family. It can be a synthetic insulin receptor ligand. The actual underlying experimental design doesn't matter as long as it involves some kind of functional manipulation of a member of the insulin receptor family. And once we've made our consensome for the purposes of our subsequent our subsequent analyses, we identify a couple of sub groups of the underlying targets. There are those that we can identify as confidence targets, which have a consensome Q value of less than 0.05, or if the consensome is large enough and we have enough underlying data sets, we can actually also designate a set of targets. Can you hold on one second? Okay, I do apologize. We can identify also the 95th percentile of the consensome, and that's what we refer to as high confidence targets. So the first kind of vignette that I want to introduce, having kind of introduced you to the kind of underlying analytics here at SPP, was where we used this kind of omics based analysis, which basically the goal here was to establish consensus around the interface between infection by SARS-CoV-2 infection and human cell signaling. And what we wanted to do was to number one is to identify targets that are most consistently induced or downregulated in response to infection by SARS-CoV-2, number one, but also to identify those specific nodes that are involved in the response. So in order to do this, we used again used consensome analysis, but here we weren't computing a consensome for a node or a node family, but rather it was a consensome for infection of a cell by SARS SARS-CoV-2. And so we surveyed over multiple independent experiments that involve infection by the virus. We ranked the downstream targets by the frequency with which they were differentially expressed, and we then identified confidence targets, and then also high confidence targets. And the results are kind of depicted here in this sort of familiar form of a volcano plot with the log FC in response to infection. So this is the amount that a particular underlying target is going to be upregulated in red, or downregulated in blue, and then we have the inverse of the log of the confidence here. So you can see here at this p-value of 0.05, we have identified confidence targets that are upregulated and those that are also downregulated. Now, the first thing that we wanted to do was to validate the consensome. And so, in other words, what we wanted to do was to convince ourselves, and equally importantly, we want to convince other people that this was going to be a reliable research resource. Now, we can kind of eyeball the consensome, and we can see elevated rankings for things that we would expect to see. For example, the role of interferon in the innate immune system is obviously very well defined, and so we saw, and we would expect to see high rankings for things that are known to be regulated by interferon. But in order to kind of rigorously and more kind of independently validate the consensome, what we needed was to kind of benchmark it against independent independent study. And one of those emerged earlier this year, and this was basically a set of nine genes, SNPs, in which were overrepresented in critically ill COVID-19 individuals. And what we found was that when we kind of overlaid this set of nine genes onto our consensome, we find that their distribution kind of skewed heavily towards these high confidence targets. And so what this told us was that the consensome was actually identifying targets that are actually relevant for clinical COVID-19. So having established that we were able to use consensome analysis to model the effect, or the effect in terms of expression of transcriptional regulation by SARS-CoV-2 infection, we next wanted to go one level up, if you will, and see if we could adopt an omics-based approach to identifying nodes that were actually regulating these responses in response to infection. And we would expect to see things that have got well-known roles in the innate immune system, but we want to go one step further and use our kind of unbiased, if you will, approach to kind of implicate other nodes. So in order to do this, we developed a new analytical pipeline, which is HCT intersection analysis, which, as the name implies, computes significant intersections, between the SARS-CoV-2 HCTs and those from our large library of high confidence targets that we have within SPP. And over the next few slides, I'll be showing the results of these intersections in the form of a scatter plot of the nodes in this library, in which the odds ratio, which is the amount over the expected intersection, increases here from left to right on the x-axis, and then the confidence of the intersection, the significance increases from bottom upwards on the y-axis. So you'll recall earlier that we designate the 95th percentile of a consensome as high confidence transcriptional targets or HCTs. And when we ran HCT intersection analysis on these SARS-2 HCTs, we found that there were very robust high confidence intersections for nodes that have got known roles in the innate immune response, and these include things like IRF1, which mediates signaling by interferon, and also STAT2. So this is what we would expect to see, based upon what's known in the research literature. But again, as with the consensome, we needed a more kind of independent, evaluation of whether or not we were identifying things that were actually involved in the response to SARS-2 infection. And in order to do this, this article, which came out earlier this year, in which the authors did a CRISPR screen, which identified nodes which were essential for SARS-CoV-2 infection. And again, reflecting the kind of accuracy or the reliability of our analysis, nodes that have significant intersections with the SARS-2 HCTs were enriched among the highest ranking hits in this CRISPR screen. And so again, what this told us, what this kind of independent benchmark was saying, that using our approach, and then using this functional infection screen, we were actually identifying a kind of overlapping set of nodes that were important for SARS-2 infection. So what so this is nice, right? So, but what new insights can we gain from this? And so I'm just going to focus down on one of these here. There are multiple ones that I could discuss here, but I will focus down on one which involves HCTs for the progesterone receptor. What we found was that there was this very nice, robust, high confidence footprint for PR among SARS-2 HCTs. And what we found was that when we looked at the kind of underlying data points, we found that a loss of function of a PR was actually had the same effect as SARS-2 infection. And so this kind of it gave rise, or it led us to kind of hypothesize that agonists for a PR might be used to actually treat COVID-19. Now, since we don't have our own lab, or we don't have access to patients, this was kind of all that we could do, was to kind of put this out there. But what actually again came out earlier this year was the results of a clinical trial that indeed showed that progesterone actually improved COVID symptoms in males. And so this was a nice endorsement or a kind of a validation of what we had predicted using our consensome. And so, in order to make this entire universe of data points available to the research community, we teamed up with the Edgar lab in UCSD, and we developed this nice Statiscape style series of networks in which users can link from the kind of underlying, or from these index networks into the actual underlying underlying information on the SPP website. And again, this work was published late last year. So the kind of second story that I wanted to walk you through here was a collaboration with Carl Betzig, and Carl's lab has developed this really nice chimeric human mouse liver model, which accurately recapitulates the accumulation of a lipid, which is a characteristic of a clinical NAFLD. And to kind of summarize, a lot of work here in one slide is that if you give these chimeric mice a 3 months of a high fat Western diet, the human hepatocytes will actually accumulate lipids, whereas the mice hepatocytes, which are side by side in the same liver, will not. And so our what we wanted to do here, or for SPP's a mandate here was to basically validate this model against networks that are known to be involved in clinical NAFLD. And so to kind of look at this in more detail, we focused on a set of 12 transcripts that were differentially expressed in response to a Western diet in both the human and the mouse hepatocytes, but whose relative expression levels were at least 1.2-fold higher in the human hepatocytes. And when we looked at these in more detail, what was really obvious was that half of these were actually encode enzymes, which are involved in the de novo cholesterol biosynthesis. And so this was interesting, and there's a kind of obvious link between cholesterol and NAFLD, but again, we wanted some kind of independent benchmark against which we could put this data, right? So what we wanted to see was just to determine whether elevated expression of these enzymes, how well does this actually reflect clinical NAFLD. And so in order to look at this in more detail, we again turn to consensome analysis, and what we did was we found a bunch of archived data sets that compared the expression between clinical NAFLD and normal liver. And again, we identified a set of confidence targets within this consensome. So I mentioned earlier the importance of validation of our analytics. And so in order to do this, we basically benchmark this consensome against a signature that was shown in this article late last year that was basically correlated with the severity of clinical NAFLD. And so again, when we took this signature and we overlaid it on the consensome, and we looked for a distribution between things that were upregulated with confidence in our consensome, we found that these were enriched within these targets. And so what this was saying to us, again, to kind of do a recap here, is that the consensome is actually working in that it identifies, or it assigns high rankings to things that are relevant to NAFLD. And what was really interesting as well, and which kind of confirmed the clinical relevance of our model, is that we found that 4 of the 6 enzymes that we had found were more highly expressed in the human hepatocytes relative to the mice ones, were actually also highly ranked within the consensome. And so this was what this was doing, was this was confirming the clinical relevance of this model. And so again, we wanted to, this was nice because what this said was that the consensome was, identifying things at the level of expression. But what we wanted to do was to see whether or not it was also identifying nodes that actually that are also relevant to clinical NAFLD. And so just to kind of refresh your memory of the HCT intersection plots that we'll be looking at over the next couple of slides, the position of a node in the plot indicates increasing confidence on the y-axis and then increasing odds ratio up on the x. So again, in keeping with the theme of validation against external benchmarks, which I keep coming back to, but this is important for us to sort of gain the confidence of users. We basically identified a study that had identified a set of 5 factors that motifs for which were enriched among targets that contained SNPs that were associated with NAFLD. And I'll refer to this study here that identified these 5 nodes by the surname of the first author, that is Namju. So just to kind of refresh your memory again, we wanted to see, we were specifically interested in things that were regulating regulating the expression of these NAFLD up HCTs in our consensome. And when we ran HCT intersection analysis on these targets, and then we kind of overlaid this Namju signature, we found that 4 of those 5 nodes actually had really nice high rankings within this HCT intersection analysis. And so again, what this was saying to us was that our mice model and that SPP analysis of this mice model actually confirmed that it was relevant to our clinical NAFLD. And we next, what we did was we then identified, we did the HCT intersection analysis on these. I'll just kind of go back here. We then did HCT intersection analysis on these 6 targets, and what we found was that we got 4 nodes, but all of which have been shown previously, or that have been linked to NAFLD. And so this was a really nice endorsement, number 1 of the mice model, but also number 2 of the ability of SPP analysis to identify nodes and targets which are actually relevant to NAFLD. Okay, so I'll just skip through these next set of slides here. So this work has been actually published recently. So if you're interested in learning more about the analysis, you can find out more here. And so the third kind of vignette that I wanted to kind of walk you guys through was actually done with one of last year's summer interns. in the lab of Dr. Olivia Osborne in UCSD. And one of Dr. Osborne's areas of interest is the mechanisms that underlie antipsychotic-induced weight gain. And what her lab is kind of interested in is understanding more why some individuals are more prone to weight gain in response to antipsychotics than others. And so to kind of study this in more detail, Dr. Osborne's group set up this mice model in which baseline blood was taken from mice. They were then given this antipsychotic olanzapine, and they were then separated out into mice that gained weight and those that were resistant. And they then took this baseline blood RNA from these individuals, and they then did RNA-seq, and they identified two sets of genes. There were those whose baseline blood expression levels were at least 1.5-fold higher in the weight gain individuals, and these were indicated as AIWGP. And then there were those whose baseline blood expression levels were at least 1.5-fold higher in these weight gain resistant individuals. And basically SPP's mandate or kind of role in this study was to basically validate this model in the context of mice lipid metabolism, and also clinical antipsychotic-induced weight gain. So we hypothesized that nodes that were upstream of the AIWGP targets would be enriched for genes whose deletion results in decreased weight phenotypes. And so, in order to explore this in more detail, we used a couple of existing resources. That is MMPC and also IMPC. And basically, what these organizations do is they map mouse genes to experimental phenotypes, and that enables us to infer what the function of that particular gene is. So we went into MMPC and IMPC, and we identified a set of genes that map to one of two phenotypes. Firstly, a decreased total fat mass, and then slow postnatal weight gain. So remember that our kind of our hypothesis was that nodes that regulated the expression of targets that are upregulated in the AIWG prone mice would be enriched for nodes that are in one of these two ontological categories. So just one last time, I'll remind you of what you're looking at here. The position of a node within this scatter plot. There is an increasing odds ratio on the x-axis, and then increasing confidence on the y-axis. And indeed, what we found was that when we ran HCT intersection analysis on the IWGP AIWGP targets, and we then overlaid the nodes that were in these categories, we found that they were actually over represented among things that had significant intersections with the AIWP or AIWGP targets, and so this was consistent with our hypothesis that things that were regulating the expression of these weight gain prone targets, when you actually knock those out in mice, they result in phenotypes that are associated with weight loss. And so finally, what we wanted to do was we wanted to evaluate how relevant this model was to human weight gain. And so what we did was we went into a previous study that identified about 150 human genes whose blood expression levels are associated with clinical body mass index. And when we took mice orthologs of these clinical BMI associated targets, and we kind of overlay them on the AIWGP or AIWGR targets, what we find is that these things that are associated with clinical BMI are kind of highly enriched among the AIWGP targets. And so what this kind of indicates to us is that there's some kind of fundamental conservation of the mechanisms that regulate weight gain in between mice and humans. So I wanted to kind of stop there and okay, so what I wanted to do now was to go through a few examples of how to use SPP. So you can feel free to kind of follow along on your own laptop. So from the home page, you've got a couple of options. You can either go into the datasets, or you can then go into OMiner. We'll start with the actual datasets. So as I mentioned earlier, there are various categories of datasets, receptors, enzymes, transcriptional factors, but also models, which I'll be discussing in a bit. So you can filter the listing based on this if you want to look at a specific category. If you're more interested in a specific, physiological system, immune system, or metabolic organs, you can also filter that way. So next, we're looking in detail at a specific, specific data set. We'll look at the transcriptomic ones first. So I'll just open this data set at a new page. And actually, that's a bad example. Okay, so what you're looking at here is basically the kind of SPP annotated version of this archived data set. Now, one of the questions earlier asked where we get these from. We get most of our data sets from gene expression omnibus, which you may or may not be familiar with it. They do a reasonable, they do a reasonably good job of kind of archiving the essential information, but kind of underlying data set isn't really that easy to use. And so what we do is we basically, identify the underlying experiments, and we identify things that are most highly upregulated or downregulated, and that's really just for people who want to have a kind of a quick look at the data set. You can toggle through the various experiments, or you can also download the data set onto your laptop. Really, all of the, or most of the essential information that you need is going to be up here at the top. You can cite the data set. You can also download that into your favorite reference manager. And then what's important, which kind of puts the article into some kind of context, is the actual original article that it was associated with, and you have a number of options here to link out for more information on those. So that was transcriptomic data sets. I wanted to also just show you for the ChIP-seq data sets. I'm going to open this particular data set in a new window here. So these we just sourced from the NCBI Sequence Suite Archive, SRA. Again, they do reasonably well in archiving the essential information, but again, reuse, the kind of emphasis here isn't on reuse or kind of ease of reuse, and so that's where we come in. We basically retrieve the kind of the underlying data sets, and we again identify those targets that are most highly ranked within a particular experiment. So that's really the purpose of this section of the site is for users to identify a specific a data set of interest. But what do you do if you want to actually use this kind of large universe of information as a whole? And so in order to do that, you would take a look through OMINOR, and as the name implies, what kind of OMINOR is designed to allow you to do is to mine this information. So again, not looking at one specific data set, but looking over all of the underlying data sets. So you have a few options here. So say you have a particular single gene of interest, and you're interested in identifying signaling nodes that regulate the expression of your particular gene of interest. So I'll begin typing here. So I will. This is IER3. This is a member of the immediate early response family. And so in order to get as much information back as possible, initially, because I want to kind of keep an open mind here, I'm going to leave all of these menus here at their default settings. And so that's going to look over all of the various categories, all of the species, and then all of the underlying physiological systems. So if you submit, what you'll get back is what we refer to as the regulation report. And what this is doing is it's looking over all of these underlying data sets, like this one, and all of the other ones, and it's actually pulling in information in which this particular target is significantly differentially expressed. So I'll just focus down here on, say, I will say IL-6 receptor family. So if you click on the data point, what you'll see is that in mouse whole liver, from mice that have been given either IL-6 or vehicle, IER3 is upregulated, and here's the p value. And so from that, you can infer that signaling through the IL-6 receptor upregulates the expression of IER3. One thing that I want to point out is that in order to kind of minimize the load on the server, when the default setting for the full change minimum is set at 2, but you can actually go in and then specify the minimum full change. And what that will do is that will return additional data points here indicated in a gray that don't make that full change cutoff, but are nevertheless significant. That's so what you're looking at here is for transcriptomics, but you can also get evidence from our CHCT experiments. And again, what you're looking at here is individual data points for IER3 in which a particular node there is evidence that a particular node might be regulating the expression of IER3. So that's the first option. So I will reset the form now. It's a good idea if you are running a bunch of different queries to kind of reset the form after each one. And I will I'll go on now to gene ontology term. And what you're looking at here is that rather than looking at a single individual target, you're looking at multiple targets that have been mapped by the Go organization to a specific specific cell function. So I'm going to say I'm interested in the cell cycle. So I'm going to begin typing. I'm going to select from the list. That's important. And again, because you're submitting multiple targets here, there's going to be extra load on the server, and so in order to minimize that, what you need to do is to select at least a specific module, category, and class. So I'm going to select receptors, and I'm going to select catalytic receptors, and I'll leave the other format menus at their default settings. And if I submit that, what you will get back is evidence for targets that have been annotated to the cell cycle that are regulated by catalytic receptors. The default view that you'll get back is the list of targets, right? So these are things that are involved in the cell cycle. But you can actually switch this up to look at the node category, and what this will then do is it'll basically rearrange your view to so that you can identify specific families that may be of interest. And so if one is of interest, you can then say, okay, I want to see more detail on EGF receptor. So what you can do is go back up. You can select EGF receptor. You can rerun the query, and what that will do is it will return data points that are specific for EGF receptor. And again, this is that kind of walkthrough was specific for transcriptomics, but you can also get the exact same information from on a CHCT information. So I'll select enzymes, and let's say I'm interested in acetyl transferases that are regulating things that are involved in the cell cycle. Now, what you'll find here is that you've actually gone over the limit of what you can see in the actual UI. So what you can do is either download these into Excel. Well, actually, yeah, that's the only thing that you can do here. So, just be aware that you can also, focus on a specific family of acetyl transferases. Well, that's also too large. Let's try NCoA. Yeah. So these are members of the CoA acetyl transferases that are involved in the regulation of targets that are involved in the cell cycle. So the third strategy that you can use is consensomes. So in order to do that, you would specify consensomes, and since we were earlier discussing insulin receptor, I will focus on those. And so what this is telling you is that this consensome is based on a total of 38 underlying experiments. So I'll submit that. And again, these are your kind of a downstream insulin receptor targets that are ranked by their confidence. And so everything up here is going to be sort of high confidence. And so if you want more information, you can look at the underlying data sets. And here, what you can see, very obviously, is that in response to all these different insulin receptor agonists, is that this particular target here is being upregulated, and so you can infer that insulin upregulates the expression of this target. Finally, the final option that you have is to upload your own list, right? So you might have your own list of targets from your own lab, say from an RNA-seq analysis or a CRISPR screen. So what we're going to do here is I'm going to, all right, so here what I'm going to, say that I'm interested in autophagy. And so I want to identify evidence for things that are involved, or nodes that are involved in the regulation of targets that are involved in autophagy. So here I've gone to JSEA, and I'm going to basically download this set of canonical targets that are involved in autophagy. So in order to upload this into SPP, I need a list of symbols, which I have here. These are the approved symbols, but they need to be in the form of a CSV. So I'm just going to specify that. Okay, so I'm going to call this autophagy, and then .cs. And then I'm going to go back into SPP, and I'm going to select the list. I'm going to choose the file that I just created, which is the CSV file. I'm going to select a specific, class of node. Here we're going to be looking at, evidence for BCA receptors that are involved. And again, you get the list of targets by default, but you can also switch to look at specific node families that are involved. So hopefully that was a useful a walkthrough for you. If any of you guys want to have a more a detailed a one-on-one walkthrough with me, feel free to email me.